already congratulated you, but I think that it doesn't will probably not bother you if I congratulate you again. So with all this, please, Jorge, I mean, you're very welcome to give your presentation. You're probably also aware of the main interest of Jorge on genomic regulation, particularly genomic regulation in beta cells, and also pancreas development, monogenic diseases. I think that these are more or less the three main topics that you cover currently in your research. So please, you're allowed to talk of whatever you want of this. I know what is the title of your presentation, but nevertheless, I mean, we're going to learn about whatever it is. So welcome, Jorge.
So I'm not going to talk about transforming translational science, I'm sorry. Okay, so thanks very much to Antonio and also to Eduard for these really kind words, and certainly to MSD for supporting this symposium. I really look forward to the discussions and also Ulf Smith's talk after this. So the topic I'm going to discuss here, because I was asked to talk about this, although I will discuss this topic from a very personal standpoint, the topic is whether we are capable to foresee, whether we have a vision of how we could develop in the future precision therapies for human diabetes. And by precision therapies, I mean this very exciting and very attractive idea of being able to target therapies to molecular mechanisms that underlie the pathophysiology of the disease, rather than simply trying to dampen the symptoms. And of course, this is something that's working really well in many diseases already, in some forms of cancer, and some neurological diseases, autoimmune diseases, where there's an in-depth understanding of the molecular defects and therapeutic approaches that attempt to address and to correct these. In diabetes, and in particular in type 2 diabetes, the most common form of diabetes, we have really an impressive arsenal of drugs that are being increasingly effective in terms of ameliorating blood glucose by stimulating secretion of insulin action, or just eliminating glucose. But in fact, none of the therapies that we're using right now has the intention to really correct the underlying molecular defect in type 2 diabetes. And the reason for that is because we don't know what the underlying molecular defect is in type 2 diabetes. So what I'm going to talk about here, and that's why I'm saying it's a very personal standpoint of how to go about this, is about how regulatory genomics can be a very powerful strategy to understand the pathophysiological molecular mechanisms of human diabetes and how these can actually provide therapeutic targets. And so in terms of genomics, what is clear is that the human genome sequence draft, which was actually declared completed in the year 2000, has produced more than 20,000 protein coding genes. And since then, there's about, more than 4,000 genes that have been shown to be mutated in many Mendelian diseases, but also increasingly in somatic mutations in cancer. And this has, of course, led to many precision therapies or to the development of therapies that attempt to target to correct the genetic derangement underlying disease. Now, of course, this is really impressive. I think we should not underestimate this, but at the same time, we know that for many common diseases, including type 2 diabetes, this has not solved the problem in terms of genetic mechanisms. And it's actually also true for many Mendelian diseases. So more than 50% of patients with Mendelian diseases, including diabetes, we don't know the underlying genetic derangement. And so, this is true on one hand, but at the same time, we now know that in addition to protein-coding genes, the genome is full of other things. So it's become apparent that it's full of non-coding RNAs. There's tens of thousands of non-coding RNAs, and there's hundreds of thousands of transcriptional enhancers. So these are essentially switches that turn on and off genes. And so the challenge right now is to really understand how these non-coding functional elements work, what genes they regulate, how they do it, and perhaps even more challenging to try to understand how defects in these regulatory elements can lead to diseases. And perhaps that could explain many of the gaps in knowledge that we have right now concerning genetic mechanisms of disease, and identify new pathways involved in pathophysiology. So I'm going to talk about regulatory mutations in Mendelian diabetes first. I would then discuss regulatory variation in polygenic type 2 diabetes, and I'll try to wrap up talking about how this could potentially lead to networks that could provide a different type of therapeutic target from what we have so far. And so since this is not a specialized audience, I'm going to go through some things that will be very basic for some of you, but perhaps not for others. And because I'm going to talk about transcriptional enhancers, I'm just going to explain a little bit what they are. They're essentially stretches of DNA, a few hundred base pairs, that are actually platforms for DNA binding transcription factors. And when they are recognized by the right transcription factors, they gain proximity with their target genes, which are sometimes very far away in the linear genomic template. But by this type of looping type of changes, they gain proximity with their target genes and promote transcription of these target genes. And the important point is that enhancer sequences, I've told you there's hundreds of thousands throughout the genome, and there's actually in each cell type several tens of thousands that are active. And depending which enhancer sequences are active in a particular cell type or developmental or physiological setting, different genes get turned on or off. And so this is, to a great extent, what determines what genes are transcribed in a particular cell type. And importantly, we can now determine what DNA sequences are behaving as active enhancers in any particular sample, because for example, the nucleosomes that flank active enhancers typically have a certain signature of modified histones. And they're also bound by certain proteins, including certain transcription factors. And so we can use chromatin immunoprecipitation coupled with sequencing to immunoprecipitate with antibodies against these modified histones, and then sequence the DNA that is bound to those nucleosomes, and can also pull down enhancer binding proteins and sequence the DNA. And map it to the human genome sequence in silico. And so at the end of the day, what we have is maps like this. What you see here is like 2 million base pairs. And you see these peaks that are stacks of sequence reads that are telling us that there are active enhancers at these locations, because they are these DNA sequences are bound by acetylated lysine 27 of histone H3 or by mediator. And so we can generate very accurate maps of active enhancers in any sample that we would like to study, in this case, for example, in pancreatic islets. And so we've generated a few years ago maps of regulatory elements in embryonic pancreas, in human embryonic pancreas. So we have the locations of active enhancers in early embryonic pancreas, and we use that for several studies. It had several interesting findings from that, but one of them was done in collaboration with Andrew Hattersley in Exeter. They were studying at the time patients who were born without a pancreas, a very, very rare disease, And they had actually found the mutations for many of the cases, but for a subset of patients that only had isolated pancreas agenesis and no malformations, even after sequencing the entire exome, they could not identify the mutations. And so what they did is they went ahead and sequenced the entire genome of proband with this disease. And to cut a long story short, this is published. Integration of whole genome sequence analysis, homozygosity mapping, and these regulatory maps told us that there was a single embryonic pancreas a regulatory element enhancer that carried homozygous rare mutations that were causing pancreas agenesis. And so these mutations all fell in one enhancer. 10 out of 12 independent probands with isolated pancreas agenesis had a homozygous mutations there. And the enhancer was 25 kilobases, 25,000 base pairs away from PTF1A, which is a known pancreatic regulatory gene. And most of these were actually point mutations in the transcriptional enhancer. And they were actually loss-of-function mutations. They disrupted enhancer activity and disrupted binding of transcription factors to their binding sites in that enhancer sequence. So this is a really rare disease, very rare disease. Pancreas agenesis, very rare cause of neonatal diabetes, which is already very rare per se. But it's interesting that the most common cause of this rare disease are mutations in an enhancer and not mutations in a protein coding gene. And so this raises the question, is now that we can sequence whole genomes, not just exomes or candidate genes, how often are we going to find this, that it's enhancer mutations rather than protein coding mutations for Mendelian diseases, right? So severe loss of function mutations, because we can now relate whole genome sequences to functional non-coding element maps in disease-relevant tissues. So there are several outstanding questions here. One is, how often is this going to happen? And to address this, and this is just an advertisement, Claire Morgan, a computational postdoc in our lab, has set out to directly address this question. She's doing a human genetic screen for penetrant islet enhancer mutations in patients who clinically have monogenic diabetes, but they don't have a mutation in coding sequences. And so what she's done is she has designed a targeted sequencing platform so that she can sequence all islet enhancers and embryonic pancreatic enhancers, or at least the most meaningful subset of these sequences for less than £100. And so this allows her to sequence well over 1,200 at this point cases, many cases with families with autosomal dominant like inheritance. And this is done in collaboration with a whole consortium of groups that have collected patients where they have screened for coding mutations and failed to find any, but nevertheless have the phenotype of Mendelian monogenic diabetes. And so it'll be exciting to find out how often subsets of this large cohort of patients have diabetes due to mutations in enhancers rather than in protein coding genes. So there's actually many other questions that we can pose by what for us is a very exciting result that has really sparked a lot of research in our lab. And one of them is really, why is it that certain enhancers, why could it be that certain enhancers harbor pathogenic mutations? Surely the vast majority of enhancers are not vulnerable to pathogenic mutations. So which enhancers are likely to be important in that regard? And how do regulatory mutations and protein coding mutations differ in terms of pathophysiology? So how are enhancer defects different from coding defects in terms of their pathophysiology? And so to try to address this, what we're doing right now is we're modeling enhancer mutations in mice. And in this particular case, the PTF1 enhancer mutations, this enhancer is very highly conserved in mice. They're not always conserved enhancers. This one is. And remember, I told you it's 25 kilobases away from the PTF1A gene. The PTF1A gene is a pancreatic regulator, so the knockout of PTF1A in mice, but also in humans, has no pancreas at all, just a very tiny rudiment. And they also don't have, they have a cerebellar defects, they have retinal defects, they have many other defects. But the mutation in this enhancer only has a pancreatic defect. They're born with a smaller pancreas. And so what Irene Miguel has done is she has deleted this enhancer. She's actually created a couple of models, but one of them she just deleted 400 base pairs that contain this evolutionarily conserved sequence that harbors the mutations in human. And mice don't always do what you want them to do, but in this case, they became very severely hyperglycemic, and the pancreas is indeed smaller. It's much smaller. It's about 50% of normal. And so why is this happening? Well, if we look at the adult pancreas, actually PTF1A, so that's supposed to be the target gene of the enhancer, is actually expressed normally in the adult pancreas that is formed in the knockouts, in the homozygous knockouts. So why does this enhancer mutation cause small pancreas and diabetes if PTF1A expression is normal? And so we had to retrace our steps in development. And before I actually explain the results, very briefly, let me just tell you a couple of brief things about pancreas development, which actually starts with 2 pancreatic buds emanating from the gut, from the embryonic gut. And these bud cells are essentially multipotent progenitor cells. And they express low levels of PTF1A. And then as pancreas development proceeds, these progenitor cells become more specialized towards duct and endocrine or acinar lineages, and eventually develop into an organ through branching and differentiation. And so what happened in these knockouts? Well, if we go to the very beginning of embryonic pancreas development, they have these 2 buds. So the knockouts have the 2 buds that are normal, but actually in the bud, PTF1 expression is not observed. So the enhancer, therefore, is required not for expression of PTF1A in the adult, but in these very early multipotent progenitor cells. And then what happens is that there is deranged differentiation and formation of these specialized duct and endocrine progenitors. And in fact, the acinar compartment, exocrine compartment, grows quite well, but there's a very severe reduction of endocrine differentiation. And so I've gone through the data actually very quickly, but essentially in a cartoon format, this is what happens. You have the 2 buds that are formed in the knockout, but the progenitor cells actually don't express PTF1A, and that results in defective formation of duct and endocrine progenitors. And so the pancreas is formed, but with a very reduced number of endocrine cells. So even though the pancreas is only 50-60% of normal, which normally, as Eduardo Montaña would probably explain much better than I can, that would not result in diabetes. It would not be expected to result in diabetes because you can live with half a pancreas, both mice and humans, without becoming hyperglycemic in principle. But in addition, they have a differentiation of endocrine cells. So this is telling us something new, as human genetics very often does, about a very specific molecular mechanism. So PTF1A in these progenitors primes endocrine progenitors. This was not known. But perhaps more generally, more interestingly, from the standpoint of what I was talking about before, it's telling us how enhancer mutations, PTF1 enhancer mutations, they don't disrupt a whole gene product. It's not like a stop codon. They just disrupt a very specific developmental program that is regulated by PTF1A of many other programs that are also regulated by PTF1A, including acinar differentiation, cerebellar differentiation, retinal cells, etc. So this is really a very surgical defect that's caused by this particular enhancer mutation. Okay, so what I've talked to you about here is Mendelian monogenic disease. And so what about type 2 diabetes, which is the certainly much more prevalent disease? What do we know really about type 2 diabetes? I already mentioned before, we really ignore the pathophysiology at a mechanistic level of type 2 diabetes. I think this is fair to say. There are some things that are known, and there's different views on this, but by and large, we don't know what's going on in humans with type 2 diabetes. We know a lot about mouse type 2 diabetes, but not very much about human, I would say. What we do know, however, is that there's more than 100 genomic loci that have variants that increase our risk for developing type 2 diabetes. And it's a really very tiny increase in risk, but very significant. And so what that's telling us is that there are variants in these loci that affect a molecular mechanism that is directly relevant to pathophysiological pathways that are deranged in type 2 diabetes. And the fact that they are having a small effect doesn't negate the fact that they are affecting a pathway, and they therefore represent a way that we have, a clue that we have as to what those pathways actually might be. And so this is how I think we should see this, and many others, of course. And so to try to understand what these SNPs are actually doing, a few years ago we generated maps of enhancers, and this was actually work done by several people, but including Lorenzo Pasquali, who's also an investigator of CIBERDEM in Canruti currently. And so what we found by generating maps of enhancers in adult pancreatic islets now, not an embryonic pancreas, is that enhancers that regulate tissue-specific activity in pancreatic islets tend to form clusters. So they don't work alone. They tend to form clusters almost invariably. And very interestingly, we found that variants that affect type 2 diabetes risk are highly enriched in these clustered enhancers. And we actually found several examples where risk variants disrupt enhancer activity in different types of assays I won't really go through. And so the bottom line from these studies is that variation in gene expression in pancreatic islets due to common variants in islet enhancers affects our risk for developing type 2 diabetes. And so this is, I think, a step forward in terms of understanding what these SNPs associated with type 2 diabetes might be doing, but of course it's not by any means the end of the story, it's just the beginning. Now we need to figure out, well, what are these enhancer variants actually doing in terms of what genes are they regulating, what is the biological impact in beta cells. And so this is where the field really is right now. We're trying to sort this out, us and many other groups. And so one approach to tackle this question of what genes are being regulated is to consider the 3D structure of chromatin. And so the idea here is that if you want to know what gene is regulated by an enhancer, you don't look at the genome browser to look at the linear genomic template. You have to see which genes are in proximity with the enhancers in the nucleus of pancreatic islets. And there's assays to do that. I won't go into the methods, but it's essentially there are variations of chromatin conformation capture in which cells are exposed to formaldehyde, so that cross-links genomic regions that are in proximity. And then chromatin is cut, religated, and so these ligation events are indications of proximity. And so we can sequence these ligation events to create maps of interactions, we call them, but they're just really proximity events between genomic regions, right? And so to cut a long story short, what we've done is we've implemented a high-throughput sequencing-based readout of libraries of such ligation events to determine long-range interactions between genomic regions in human pancreatic islets. And we've done this with a method called promoter capture high-C that's been developed by Biola Javier and Peter Fraser from Abraham Institute. Well, Biola was there at the time. And they developed this method which allows very high-resolution mapping of interactions that involve promoters. So this is the sort of maps that we have. Wherever you see an arc, this means that this genomic region is in proximity with that genomic region, even though they're far away in the linear template. And so here we have these maps from pancreatic islets, but other people have created maps in other tissues. And so we can determine which interactions are actually specific to pancreatic islets. And we've learned a lot of interesting things I will just go through rather quickly. One is that we have, there's these clusters of enhancers I was telling you about before. There are, with these 3D maps, we know that there are hubs of enhancers that are three-dimensional hubs that very often target more than one gene. And what's interesting about these hubs of enhancers is that they're very highly enriched in type 2 diabetes associated variants, more so than if we just look at these linear maps. And so enhancers in the hubs are much more enriched than enhancers outside of the hubs. And furthermore, they are enriched in type 2 diabetes variants that have p-values of significance for association with type 2 diabetes, not just above the threshold for genome-wide significance, but also below. So we believe that in aggregate, variation in these hubs makes a very significant contribution to type 2 diabetes risk, even though each individual SNP has a very tiny effect. And so what's important is that we can use these maps to actually assign target genes to enhancers. So we know if an enhancer carries a variant associated with diabetes, what is the likely gene that's going to be affected by that variation. And so here's an example of an enhancer, which is in a cluster. So here, just to put the context, this is a genomic region of a couple of hundred kilobases. And you have here a cluster of enhancers. Here is a type 2 diabetes associated variant interval, so many SNPs associated with type 2 diabetes in this region. One of them falls right in an enhancer that is active in pancreatic islets. And so to assess what is the likely target gene of this enhancer, Ines Ebola has deleted, using CRISPR, this enhancer in a human beta cell line, now not in mice, and asked, well, what is the target gene, right? So which gene is affected by deletion? Is it this gene on this side or on the other side? And in fact, none of the two genes in proximity are actually affected by deletion of this enhancer. Instead, if we look at the interaction maps, there's another gene that's about 200 kilobases away in the linear map that is, however, in proximity in these 3D maps. So it's interacting, let's say, quote unquote, with the enhancer. And so this gene over here, which is actually a very strong candidate gene because it's been implicated in beta cell biology through a variety of different lines of evidence, including knockouts in pharmacology. This gene and the next door gene are actually show reduced expression in the same knockout cell lines. And so it's about proximity in the nucleus in 3D space, not about proximity in the computer screen, which is a linear template that matters. And so this means that we can now assign target genes to enhancers that carry variants associated with diabetes. And we can build maps of these hubs that are strongly enriched in type 2 diabetes associated variants and that are very important for regulating cell-specific genes. And so just some final words. I think I have time for 3 or 4 more slides? Yeah. Thanks. So just some final words about how can we bring this to thinking about therapies for diabetes. So the idea is that it's not about changing these alleles, which have a tiny effect. It's about experimentally modifying cells, editing these alleles so that we can evaluate how they impact beta cell pathways. And by doing this, we can identify pathways that can be targets for therapeutic interventions. And in this regard, it's important to bear in mind that we now know there's about 30 genes that are mutated in monogenic diabetes. And a majority of them are genes that are important for pancreatic beta cells. So there's some that are important in immune mechanisms or in insulin action, but the majority of them are important in pancreatic beta cell. And about half of those, of the ones that are not the immune and insulin action genes, are actually targeted by type 2 diabetes associated variants. And so what this means is that we have very different phenotypes, type 2 diabetes, monogenic diabetes, very different types of genetic mechanisms, polygenic variants or severe loss of function mutations mostly. But they all converge on the same genes. Well, not all of them, but they very often tend to converge on the same genes. And in some cases, we know that they are interrelated, functionally interrelated, and appear to form networks. In some cases, very clearly do so. And so we think that this genetic screen that I was telling you that Claire Morgan is developing will help expand this network and also help us understand the interactions between components of this network. And I think that the fact that both polygenic and monogenic forms of diabetes are telling us that these genes are so important for human beta cell function and glucose homeostasis is really telling us that these networks are really central to pathophysiology of diabetes. And so I think it makes sense to propose that trying to target these networks, not necessarily the genes that have variants in particular individual, but the networks themselves could be a new type of therapy that really aims to modify the mechanistic underpinnings of type 2 diabetes rather than simply trying to improve blood glucose by stimulating secretion reaction or other ways. So I'll leave it there, and I'll thank you for the time. I have to, well, there's actually many people I have to acknowledge that have been doing the work that I've been talking about here, including Irene Miguel and Miguel Angel Maestro, the PTF1 enhancer knockout studies, the promoter capture high-C, several people in different angles, including Ines Ebola, I already mentioned, the genome editing, Delphine Rolando, and in the analysis, as well as Joan Ponsá and Silvia Bonas in the type 2 diabetes enrichment analysis. And I have too many collaborators here to mention all of them. So thanks very much. I'll be happy to take questions.